Okay, welcome everyone. David Hafler from New Haven, Connecticut, Yale School of Medicine. I'd like to welcome you all to the workshop today on the Neuroimmunology of MS, sponsored by the Prada Foundation and the Preserving the Brain effort from the foundation. Today we have four speakers. First will be myself, giving an overview of multiple sclerosis. Second, we'll have Professor David Pitt, who will discuss the pathology and genetic pathologic interactions. Then we'll have Professor Sumida, who is in our department, who will discuss some very new data related to why there's loss of immune regulation in MS. And finally, we'll have Professor Erin Longbrake discuss clinical trials and where we are with treatment of multiple sclerosis. So that will be the outline. We each speak for approximately 25 minutes. And then we will have time for questions at the end of the session. So let me start with my presentation. So again, the title of my presentation, sorry, let me just get out of this. Stop. Okay, so the title of my presentation is an overview as to the cause of MS. Now, I would argue that one never truly knows causality, but we've gone a long way in our understanding of the immune system and autoimmunity. And we have a very good working model based on the genetics, ex vivo human studies, the mouse models, and most importantly, clinical trials to suggest that we have a very good working model as to what causes multiple sclerosis. So today I'll first present an overview of autoimmune disorders and show that they're complex genetic diseases where genetic variants map to the immune system. In MS, B cells, we believe, drive myelin-specific CD4 cells into the central nervous system. There's loss of regulatory T cells, and I'll briefly talk about the background of regulatory T cells, and then Sumida will present recent data as to the mechanism of why the immune system is dysregulated in multiple sclerosis. I'll present data on T cell traffic between blood and the CNS, show that it's tightly regulated, and show a very surprising finding related to healthy subjects that in normal spinal fluid, a T cell surprisingly acquire an inflammatory gamma interferon or Th1 signature. I'll show unbiased single-cell RNA sequence analysis, which revealed novel immune pathways that we believe drive the onset of MS. And finally, one can see this inflammatory signature in healthy brain. So first, the background on the genetics of MS. So what are the genes that cause MS? Well, there are two types of genetic disorders. One related to gene knockouts. An example would be muscular dystrophy. The dystrophin gene is knocked out, and if that gene is gone, then the child will develop muscular dystrophy and muscle dysfunction. More common diseases, including the autoimmune diseases, such as MS, lupus, type 1 diabetes, are not due to genetic knockouts of one gene. Instead, they're complex genetic syndromes with multiple genetic variation, each with a phenotype. What's interesting, and I won't have time to show the data this morning, whereas each of these genotypes have a very small association of risk, the phenotypes are quite striking associated with each genotype. These phenotypes interact with the environment, including the microbiome, diet, and other exogenous exposures, which then lead to multiple sclerosis. Back in 2000, after performing a sabbatical with Eric Lander, we formed the International MS Genetic Consortium. I want to particularly acknowledge Alistair Compton, Steve Hauser, and Eric Lander, who together formed this consortium, which led in 2007 to the first identification of genetic variants associated with risk of developing MS, where beside MHC, we discovered the IL-2 receptor and IL-7 receptor were associated with disease risk, again, with relatively small odds ratios. The consortium has since continued investigation. In 2019, published a landmark paper where we identified 233 genetic variants associated with disease risk, examining 47,000 patients with MS, 68,000 controls. Uh, these variants, then it raises the issue of how do these genetic variants actually cause the disease. A number of years later, in about 8 years ago now, at the Broad Institute, working with Brad Bernstein, Kyle Farr, and Alex Morrison, we performed genetic and epigenetic fine mapping of the causal immune variants. Here, what we did was to take each different cell type, be it brain cells, different immune cells, different tissues, be it water mucosa, fat tissue, liver tissue, and identified where there was open, there was open chromatode as defined by K27 acetylation marks. Because we argue that if a genetic variant of the SNP was located in a region where the chromatode was closed, that SNP would not influence that cell type. But if every place there was a SNP, there was open chromatode, again, it's identified by these K27 acetylation markers, and that would suggest that that SNP is working in that cell type. Our lab generated the data related to different immune populations. At the bottom, we have different diseases, neurologic diseases, autoimmune diseases, and other systemic diseases. We concentrate on multiple sclerosis here. The red is a p-value 10 to the -30th. One can see that the genetic variants are hitting immune cells, in particular Tregs, different T cell populations, Th1 and Th2 cells and CD8 cells. Not only that, the genetic variants were specifically hitting B cells. There are only two other autoimmune diseases where the B cells are being hit, systemic lupus and primary biliary cirrhosis. So it suggested that the B cells play a critical role in multiple sclerosis. Of course, the landmark studies led by Steve Hauser, published in New England Journal in 2017, looking at B cell depletion, showed a rather remarkable effect of ocrelizumab, an anti-CD20 monoclonal antibody, in two separate studies, decreasing the mean number of MRI lesions per patient by 98%, 97%, and 96 weeks. So do we know the cause of autoimmune disease, that is, of multiple sclerosis? Well, here's the working model. That it's a genetically mediated autoimmune disease initiated in the periphery by T cells, B cells, and macrophages that traffic into the central nervous system. That microglial antigen cross-reactive with myelin, which has been amply demonstrated many times in an activated cell, can lead to secretion of inflammatory cytokines with expression of co-stimulatory molecules, leading to the activation of autoreactive T cells. Perhaps EBV infection of a B cell could be the initial event, which would trigger cytokines and co-stimulatory molecule expression. Now, this occurs in healthy subjects, but we know that they're regulatory T cells, and I'll show some data on that in a moment, that prevent the activated T cells from causing disease, and there's health. And here's data again. Dr. Sumida will discuss this in much more detail in the third talk. But back in 2004, our lab showed that there's a defect in regulatory T cells. Here's presents suppression of proliferation, patients with MS, healthy age-matched control. These are all untreated MS patients. You can see this marked decrease in regulatory T cell function. Margot Dominguez-Vera in our laboratory went on to show that it's associated with gamma interferon secretion and cytokine secretion. That is, these Tregs in patients with MS are activated. They're FOXP3 positive, but they're secreting gamma interferon in a much higher amount. And this has been a very common observation across different autoimmune diseases, including type 1 diabetes, that the Tregs become dysfunctional. So what is the transcriptional circuitry driving? Can we define a master regulator? And again, Dr. Sumida will discuss these exciting new findings related to loss of immune regulation, not just in MS, but in autoimmune disease. So here we have a picture where you have activation, microglial activation, perhaps EBV, activation of antigen-presenting cells in multiple sclerosis. In health, you have these regulatory T cells, but the myelin-reactive T cells are secreting IL-10. That is, a number of years ago, we found that there are autoreactive T cells in patients with MS, but they're also found in patients in normal, healthy subjects. in work done by Will Cow in our laboratory, went on to study this in great detail, and you can show by single cell analysis of monoreactive T cells that the T cells in MS are secreting inflammatory cytokines, in particular IL-17 and GM-CSF, similar to the EAE model, whereas healthy controls are making an anti-inflammatory cytokine IL-10. So to summarize, we believe that B cells appear to be the antigen-presenting cell, perhaps by EBV infection, drive myelin reactive T cells, but loss of regulatory T cell function, leading to the activation of CCR6, CXCR3 positive T cells, migrate into the central nervous system, secreting inflammatory cytokines. So that's a working model. And the genetics of MS, we know, hit B cells, and perhaps how viruses such as EBV insert themselves in the B cells are driving the disease. So we wanted to look at how T cells traffic into the brain. And we know that T cell traffic between blood and CNS is very tightly regulated. This is recent work led by Jenna Papalardo, a very talented graduate student in our laboratory, examining the transcriptomic and clonal characterization of T cells in the human central nervous system using single cell analysis. In this analysis, we performed spinal taps in blood for both patient and patients with MS, isolate the bloods and pellet the spinal fluid, do 10x single cell RNA sequencing, and examine each individual cell. The beauty of this technique is there's no underlying hypothesis. That is, we don't have to make any assumption as to what is there, and allows us to generate hypotheses as to the functionality. We know that this restricted immune state in the central nervous system, very tightly regulated. There are many different types of populations of T cells, but with T cell traffic through the choroid plexus, very specific population of cells get in, and a condition in the brain. I'll show that the T cells that get in acquire a T-beta gamma interferon signature. Just as an overview of what I'm about to show you, we believe this is mediated by astrocytic and homeostatic communication. Ben Barres, the late Ben Barres, showed that astrocytes condition microglia, and we believe that the astrocytes may be performing homeostatic communication, shaping not only the microglia, but T cells require a certain function. So that's what I'm going to plan to show you over the next few minutes. So we performed single cell analysis. We found 9 clusters of T cells in healthy spinal fluid. Let me back off by saying we first started examining MS patients' blood and spinal fluid. We found that the MS spinal fluid was very inflamed. You're not surprised to hear that the spinal fluid in MS was inflamed. But Jenna said, how do we know that's MS specific? And we enrolled 12 healthy subjects, healthy grad students and postdocs, to have spinal taps so we can examine what does normal spinal fluid look like. So this is looking at a heat map, doing a shape-based gene cluster, a technique developed by Sumida Krishnawami, from normal spinal, again, this is normal spinal fluid. We use a tool that Sumida developed called MAGIC. So we imputed the gene expression score based on patterns of the tissue score, can also be detailed in the paper. And we found 9 clusters based on the timing and magnitude of the tissue score. We're particularly interested in the Th1 cluster, that is a cluster of T cells in normal spinal fluid expressing the transcription factor for gamma, TBX21 or TBET, gamma interferon, granzyme K, and EOMs, all associated with gamma interferon. So looking at these clusters in greater detail, we found 3 different populations of T cells in the spinal fluid. Here's naive blood, memory blood, CSF1, 2, and 3. So the Th1 cluster is a Th1 cluster in memory CSF3. There also this cluster also in the examined, the CSF3 had markers of tissue residence like CD69 and markers of cytotoxicity. So it's highly effective gamma interferon secreting population in normal spinal fluid. Similarly, in CD8 cells before found markers of tissue residence and cytotoxicity. So to summarize, just many experiments and petabytes of data, there's a population of cells that are excluded from the tissue in the blood. There are cells enriched for entry into the spinal fluid, and there are cells that enter into the central nervous system and acquire tissue-dependent changes. If we examine in more detail in the spinal fluid, there are Th1 genes, markers of resident T cells, those involved in cholesterol homeostasis, not surprising, considering the high amount of cholesterol in the brain, TGF-beta pathway, and high amounts of co-inhibitory receptors. So we then wanted to actually look at, do we see gamma interferon screen T cells in normal spinal fluid? So here is 3 separate experiments from spinal taps of healthy subjects looking at the co-inhibitory molecule PD-1, gamma interferon on this contour plot. This is looking at spinal fluid compared to blood. So this is the potential to make gamma after 4 hours of stimulation. And one can see this marked secretion of gamma, high expression of PD-1 in spinal fluid, which is not seen in blood. Now, this is interesting because we often discuss the concept of immune privilege in the brain, that T cells in the brain are non-responsive. So we wanted to examine the phenotype of PD-1 high T cells in healthy subjects. Work was published recently, and one can see if you take T cells that express high amounts of PD-1, load them with a green dye, then stimulate them, they do not enter cell cycle. Whereas the normal PD-1 negative cells do, they dilute out the green dye. So this says, this phenocopies the so-called immune privilege in the brain, that the T cells expressing high PD-1 do not enter cell cycle, but they do make gamma interferon. Here, 50% of the T cells, PD-1 high, are making gamma but don't enter cell cycle. So we believe that PD-1 high, gamma interferon secreting T cells represent the immune privilege in the brain, which may have some biologic functionality we'll get to at the end. So now we know that the normal spinal fluid has inflammatory signature. What about multiple sclerosis? So by doing the unbiased single-cell RNA-seq analysis, we revealed a number of novel pathways we believe drive MS. First, we look for a tissue for specific cell population, the MS blood and healthy CSF and blood. And as previously reported, we did not see any difference. Here are fate maps between MS and healthy controls. The populations appeared to be the same. But looking very carefully, we could find a number of differentially expressed genes. These are log-fold changes between MS and controls. So here we have different populations. Here are the different CSF populations, C8 cells and memory cells. This is the log 10 Q value between MS versus healthy donors. Let me highlight some differences. We found major differences in MALAT1, which is an epigenetic modulator of gene expression and regulates cell mobility, comparing MS versus control, both in blood and in spinal fluid. Found differences in IL-32, which we commonly see occurring in MS, particularly in the CSF populations. It's a pro-inflammatory cytokine which induces TNF-alpha and IL-6. Found increases in the AP1 transcription factor FOS and Jun in the memory T cell in the CSF populations. So these data present an unbiased view of what appears to be the molecular mechanism driving MS, and will take us years to work through the mechanism for each of these different factors. Here's another way of seeing these data. We found that the cells that were clonally expanded in MS were more differentially expressed. So here's descending order of log-fold differences between MS and healthy controls. In the expanded versus non-expanded population, can we see high amounts of IL-32 in the expanded T cell population? Also see the Jun B and MALAT1. In the CSF3 population, we similarly see a number of different granzyme genes, TNF receptors, and in the memory CD8 population, similar genes. So what about brain? Dr. Pitt will discuss the work he's doing on nuke-seq analysis of MS brain. But just to point out, in the normal brain, we also see this inflammatory signature. So here we characterize T cells in human brain parenchyma, work we did with Professor Jack Cantel in Montreal, supplied tissue. We see the different clusters, and we see this of different populations, a small population of T cells over here. We look at the T cells, both by single cell RNA sequencing and nuke-seq, we see is a marked induction of gamma interferon and granzymes, very much overlapping what we see in the spinal fluid. So this Th1 signature we see in the spinal fluid is also reflected in normal brain. And here's this gamma interferon signature. So in summary, the fundamental question is there selective migration of Th1 cells, or are they shaped in the central nervous system? Is there glial T-cell communication circuits, as I discussed at the beginning, as first identified by Ben Barres, where TGF-beta cholesterol driving microglia function. We know that astrocyte-derived TGF-beta and cholesterol are required for microglia survival, and that soluble factor produced by astrocytes, including cholesterol lipoproteins, are found within the spinal fluid. So I won't show the data for lack of time, but it's in the paper. In fact, if you take cholesterol and expose normal CXCR3 positive T cells, we are able to phenocopy what we see in the spinal fluid. And also looking at T cell receptor sequences, we find that T cells that are in this blood with identical T cell receptor sequence to what's in the spinal fluid are remarkably different. So we believe that this isn't selective migration of activated T cells in the blood, but instead activated T cells that migrate into the central nervous system acquire this signature, this conditioned signature in the tissue based on exposure of factors from the astrocytes. So in summary, autoimmune disorders are complex genetic diseases where genetic variants map to the immune system. In MS, B cells drive inflamed myelin-specific CD4 cells into the CNS. G cell traffic between blood and CSF is tightly regulated. In healthy spinal fluid, T cells acquire a Th1 signature, and T cells in blood and spinal fluid are functionally different, indicating that CNS shapes the homeostatic T cell state. There's a greater phenotypic difference between healthy and MS in the spinal fluid, the more expanded cells, suggesting that the T cells encountering antigen undergo clonal expansion and driving much of the difference that we see. And finally, the Th1 signature is seen in healthy brain. We speculate that this Th1 gamma interferon screen T cells may be involved in physiologic processes in terms of synaptic pruning by microglia, and may have a role, even though they're rare, in trafficking through the brain, particularly at night during sleep, inducing microglia to lead to synaptic pruning and regulating neurodevelopment. So let me end by acknowledging all the individuals involved in these studies, particularly the single cell analysis led by Jenna Papalardo. I acknowledge others along the way who were involved in the earlier work we did, such as Will Cow and Margot Dominguez-Villar. Also thank David Van Dyke and Sumida Krishnawamy for the help with the critical analysis, the spinal fluid, and our MS center. And here's our MS group. I invite all of you to this some beautiful Connecticut. This is where we have our lab retreat. This is the Long Island Sound. This is where we have lab meetings whenever we can. So thank you for your time. And then I'll stop sharing. And now we'll move on to the next talk. Again, we each ask each speaker to talk for 25 minutes. That's now 24 minutes. And that will give us time for questions at the end. So David Pitt is in the Department of Neurology, Yale School of Medicine, and is trained both as a neurologist, sees MS patients, and is a neuropathologist. So David, you're on.
Okay. Thank you very much, Professor Hafler, and thank you to the Prada Foundation for inviting me. I would like to give present a different aspect of multiple sclerosis, and that is the contribution of astrocytes to MS pathogenesis. And I want to exemplify that with two different stories. One, the role of astrocytes in genetic MS susceptibility, and then a possible role of astrocytes in progressive MS. Starting with the role of astrocytes in genetic MS susceptibility, I want to start with the GWAS studies. Dr. Hafler mentioned them already. This was a gargantuan effort. Over 47,000 MS patients were enrolled and close to 70,000 controls. And the question that was asked was very simple, and that is, what is the genetic difference between people who have MS and people who don't have MS? You saw this picture here already. This is the latest view. You see here all the different risk variants. You see on what chromosomes they are. You can see the genes that they potentially perturb. You see here the significance of their effect. So what about these risk variants? Well, they are highly significant, but they confer very small effects. Dr. Hafler mentioned that already. So the odds ratio of these risk variants are something between 1.05 and 1.15 or so. So it is really small. 90% of these variants are in non-coding regions, and that makes it somewhat difficult to determine the genes that are perturbed by these variants. One approach that has been taken was that the gene that is next to the SNP is declared to be dysregulated by the SNP, but this is a very crude approach. 60% of these risk variants, they map to immune enhancer regions. So there's definitely a immune flavor to this. And importantly, there is overlap in terms of risk variants with other autoimmune diseases such as inflammatory bowel disease or type 1 diabetes. And this provides another argument, if any more arguments are needed, that MS is really an autoimmune disease. So one important question in this field is what cell types are dysregulated by these risk variants, or in a different way, what cell types confer this genetic MS risk? And you saw this picture here already. There was a seminal study by Farr in 2015. And as Dr. Hafler said, they looked at 21 different autoimmune diseases and also other diseases such as Alzheimer's disease or migraine or asthma. And they mapped the risk variants for these different diseases onto chromatin from different cell types. And the idea was, as was said before, that if a risk variant falls into an open chromatin region, then this risk variant probably affects the function of this particular cell type. And this here is a heat map where you can see where in which cell types the risk variants enrich for different diseases. So what about multiple sclerosis? You can see here that that all the risk variants essentially enrich in T cells and in B cells. So a unsuspecting reader of this paper might walk away thinking that all the risk variants affect lymphocytes, but not any other cells, for example, brain cells. So that MS is really a disease of lymphocytes. But if you look more carefully, you see that the cell types that were examined were really T cells and B cells. All the other factors here are really organs rather than individual cells. So, for example, in the brain, they looked at different parts of the brain, different lobes, different areas. But in this paper, the authors didn't look at individual cell types. So this is a very different type of analysis that you can't really compare. So while it is true that MS risk variants, they enrich heavily in T and B cells, it is still not clear whether risk variants also enrich in brain cells. And we set out to examine that. And we focused on one particular risk variant, this risk variant here. This risk variant is part of a haplotype block that is in the intergenic space between NF-kappa B1 and MANBA1. And this risk variant has been examined already in Dr. Hafler's lab in lymphocytes, their effect on lymphocytes. So we were interested in whether this particular risk variant also affects astrocytes. Why astrocytes? Because our lab is interested in astrocytes, but also because astrocytes are the most common glial cell type, and they have arguably the biggest functional portfolio, if you will. They're kind of the Swiss Army knife of the brain. So we went to our MS clinic, and portion of our MS patients are genotyped, and I selected patients that were homozygous for the risk variant, and patients that were homozygous for the protective variant. And this is a common risk variant. This is a common variant. So about 18% of the population is homozygous for the risk variant, and about 22% of the population is homozygous for the protective variant. We took skin biopsies from these patients, we grew fibroblasts, and we generated iPSCs, induced pluripotent stem cells, and these iPSCs were differentiated into astrocytes. We had, so I'm sorry. So here, oh yeah. So we then had really two cultures, cultures with astrocytes that had the risk variant and cultures with astrocytes that didn't have the risk variant. And we looked first at NF-kappa B protein expression because this risk variant is close to the NF-kappa B gene. And we found that at baseline, there was actually a difference in expression of P50, which is a component of NF-kappa B. And after stimulation of astrocytes, the difference in expression was even bigger. This was true for P50 and P65, another component of NF-kappa B. We then looked at NF-kappa B signaling, and we found that an inhibitor of NF-kappa B signaling was more downregulated in astrocytes with the risk variant, meaning there's more NF-kappa B signaling. And we found that phosphorylation of P65 was upregulated in astrocytes with the risk variant. So that would indicate that astrocytes with the risk variant have increased NF-kappa B signaling. We next looked at genes that were downstream of NF-kappa B that were targeted by this transcription factor. And we did an assay consisting of 84 different genes that were affected by NF-kappa B. And we found that a small fraction of that, in total of 9 genes, of which we show 5 here, were affected. These genes were IL-15, I apologize. IL-15, this is a cytokine, then an adhesion molecule, complement component 3, and 2 chemokines. Other activation markers of astrocytes that are not NF-kappa B dependent, like GFAP and NOS2, were not differentially regulated. And this was true for genes and for the protein itself. We then next looked at a different important function of astrocytes, and that's glutamate uptake. And we found that glutamate uptake is strongly reduced after stimulation of astrocytes. But in the astrocytes with the risk variant, there was a slight but significant further decrease in glutamate uptake. And that might perhaps initiate glutamate excitotoxicity. We then looked at another function, lactate secretion, and we found again that astrocytes with this risk variant had reduced lactate secretion, arguing or indicating that there might be neuronal astrocyte metabolic uncoupling, although glucose utilization was the same in both groups. So to summarize this, we found that with this particular risk variant in astrocytes, NF-kappa B expression was increased, NF-kappa B signaling was increased, complement component 3 expression was increased. And at that time, complement component 3 was thought to be a marker for a neurotoxic type of astrocytes. They are termed A1 astrocytes, although in recent papers, this has been, some doubt has been cast on that. But potentially, this is a marker for neurotoxic astrocytes. We found that there's metabolic uncoupling between neurons and astrocytes, and we find that there's slightly impaired glutamate uptake, which would argue that there might be glutamate excitotoxicity. In addition, we find that 2 chemokines are upregulated, an adhesion molecule is upregulated, and a cytokine that stimulates lymphocytes is also upregulated. So overall, there is a theme of lymphocyte recruitment and lymphocyte activation and a theme of neurotoxicity. So we wanted to test whether this also holds true in MS brain tissue. And I have a brain bank that I maintain in my laboratory, and we genotyped our MS cases. And we found, in fact, 5 cases that were homozygous for the risk variant and 5 MS cases that were homozygous for the protective variant. And we looked at active lesions within these cases, I think 12 lesions per group. And we stained then these lesions with GFAP, a marker for astrocytes, and other markers. And we found that in astrocytes, in reactive astrocytes, that P50 expression was substantially increased in cases with the risk variant compared to cases with the protective variant. And then we stained with all these other markers that we found to be upregulated in our cell culture. And it turns out that all these markers were in fact upregulated in risk variants with the protective variant. Markers that we didn't find to be upregulated, like GFAP or INOS, they were also not upregulated in brain tissue. We then looked, we tried to characterize these lesions some more, and we looked at lymphocytic infiltration. And we found, in fact, that there was a difference. We focused on the perivascular space, which is where most of the lymphocytes accumulate. And we found that in lesions from cases with the risk variant, that there was substantially more lymphocyte infiltration than in cases with the protective variant. And here you see the quantification. And students who analyzed these lesions noticed that there was also a difference in lesion size. So we quantified the lesion size, and we found, in fact, there was also a difference between cases with the protective variant and cases with the risk variant. The cases with the risk variant had a higher overall lesion size. Histology is not the best way to analyze lesion size because you often cut through these lesions. So we went to MRIs, and we looked at MRIs from 35 patients that had the protective variants and 41 cases that had the risk variants. And we quantified the overall lesion load, but there was no substantial differences between the two groups. So to summarize this, we went with one particular risk variant, this risk variant here. We showed that this risk variant changes the phenotype of astrocytes in culture. We then went to MS lesion tissue, and we found that the same changes that we found in cell culture were also present in astrocytes in these lesions. And we found that the lesion phenotype was changed. In fact, that the lesion size was bigger and that there was a higher infiltration with lymphocytes. We didn't find that the overall lesion load was different. And somehow this is not surprising because this is really only one risk variant out of 233. And the odds ratio that this risk variant confers is 1.07 per allele. So it's really a small effect. But this speaks to perhaps a bigger theme. And this bigger theme is that genetic risk variants, they drive dysregulation of the peripheral immune system for sure, but they also drive dysregulation of resident CNS cells. So maybe then there is a bidirectional, this is a bidirectional process where lymphocytes infiltrate the brain, try to infiltrate the brain, and brain cells, they might either prevent or they might provide access. In the case of this particular risk variant, so there are more adhesion molecules expressed that make it easier for lymphocytes to enter into the brain, and then these lymphocytes are stimulated by astrocytes. And this might make a difference between an activated lymphocytes wandering into the brain and wandering out again, or a lymphocyte wandering into the brain and causing inflammation there. And this theme that the target organ or that the targeted cells contribute to inflammation in autoimmunity is getting more and more to the fore. So for example, it has been shown that oligodendrocytes, a subtype of oligodendrocytes in MS lesions, express MHC class II. That means they stimulate lymphocytes and they probably get damaged by lymphocytes. And we recently published a paper on type 1 diabetes where we showed that the beta cells in the islets, that they express pro-inflammatory factors. And if these pro-inflammatory factors are knocked out in mice, then the mice don't develop type 1 diabetes, or at least delayed. And there are other examples, for example, rheumatoid arthritis, where the fibroblasts in the synovia express inflammatory markers that kind of keep this inflammation going. So we are now working on the question, what other MS risk variants affect astrocytes? And we have done a TAC-seq on astrocytes derived from MS lesions. And we now do the same thing like in the FAR paper that I presented before, that we look at the risk variants for MS and we map them onto chromatin of these MS-derived astrocytes, and then identify the risk variants that fall into open chromatin. And we have also identified the EQTLs of astrocytes, so we can determine what genes are really affected by the risk variants that are active in astrocytes. So this is ongoing work. Okay, I want to talk now about the second story, and this is astrocyte network dysregulation in progressive MS. This is ongoing work in our lab. And all this starts with Laura Airas. Laura Airas is a collaborator. She's in Finland, and she does PET imaging of MS patients. And she's interested in adenosine 2A receptors. And she, in this study, has given A2A receptor ligands, radioligands, to MS patients. And in addition, she has given TSPO radioligands. And TSPO, as you know, is a marker for activated myeloid cells and astrocytes. So by looking at the TSPO binding, she can identify different lesion types. And she focused on chronic active lesions. And here, as a reminder, this is how a chronic active lesion looks like. So here's the lesion rim that is characterized by inflammation. There's the lesion center that is devoid of inflammation. And here is the normal appearing white matter that is myelinated, but otherwise it's not really normal. You see lots of glial activation. And she found that in these chronic active lesions that the A2A receptor was expressed in the normal appearing white matter. And she further found that in chronic active lesions from patients with progression, the A2A receptor expression was higher than in chronic active lesions from patients with relapsing remitting MS. And that by itself is a remarkable finding, that the composition of chronic active lesions differs depending on the context, progression versus relapsing remitting MS. So what does adenosine 2A receptors, what do adenosine 2A receptors do? The natural ligand for this receptor is adenosine, and that is a stress signal. So if cell membranes are compromised, ATP leaks out into the extracellular space. ATP is then degraded eventually to adenosine, and adenosine binds to this receptor. A2A receptors are expressed by a number of different cell types. So, for example, immune cells. In immune cells, they have an anti-inflammatory effect. So they're in fact immune checkpoints. And my understanding is that immune checkpoints are being developed, as A2A receptor inhibitors for treating cancer. In the brain, it is highly expressed in neurons in the striatum. And in the striatum, they are co-expressed with dopamine receptors. And in fact, they inhibit A2A receptor signaling inhibits dopaminergic signaling. So there's currently on the market an A2A receptor antagonist that is given in Parkinson patients to increase the amount of dopamine signaling. A2A receptors are also expressed in astrocytes and in microglial cells and in endothelial cells, typically in activated cells. And there was a very nice paper that, for example, an A2A receptor knockout specifically in astrocytes improves memory of mice that were induced to develop AD. In EAE, there is a dual role. So if you give A2A receptor agonists early on in the disease, you dampen the disease. And this is consistent with the anti-inflammatory role that A2A receptor signaling has on immune cells. But if you give it in the chronic phase, then the A2A receptor agonist exacerbates the disease. And it is not entirely clear why that is. Possibly this has something to do with glial cells. So we now stained chronic active lesions just to look where the A2A receptor is expressed in MS lesions. And you can see here that at the lesion rim, there is some expressed A2A. This is this brown staining. The purple staining are activated microglial cells and macrophages. And if you go further out into the lesion perimeter in the normal appearing white matter, you see that there's a lot of A2A receptor expression, and this is all inactivated. This is all inactivated astrocytes. In the lesion center, there's barely any expression, and there's also no expression in white matter from control tissue. So we wanted to ask, so what are the astrocyte subpopulations really that express A2A receptor? And what impact does A2A receptor signaling has on astrocytes? And might this play a role in progression? And we went back to these single nuc-seq data sets that were recently generated from MS lesion tissue. There were a number of high-profile papers that generated these single nuc-seq data sets, and here's an example. But we didn't find ADORA2A, which is the gene for A2A receptor represented in astrocytes or in any of these. They were not present in astrocytes, and many other proteins or genes that we know are expressed in astrocytes were not present. And I don't want to go too far into this, but it is very clear that the astrocyte nuclei are underrepresented in these single nuc-seq data. There is a bias, and here is an example. So these are nuclei obtained from MS lesions, and you can see that the vast majority of nuclei are oligodendrocytes, and keep in mind these are demyelinated lesions, and neurons. Keep in mind that these are really white matter. That this is white matter tissue. So you see that the astrocytes are really underrepresented. They should be more than the oligodendrocytes really, and other immune cells and macrophages and microglia are also really underrepresented. Current technology, 10x genomics technology, has still a low transcript capture. This might also contribute to not finding ADORA2A in astrocytes. And then, of course, there's no guarantee that you have, if you have high protein expression, that this means that you really have high RNA expression. So in order to get some context where this A2A receptor, where the A2A receptors are expressed, we went to a different technology, to highly multiplexed imaging. And highly multiplexed imaging, with highly multiplexed imaging, you can put 20 or 30 or 40 antibodies onto a brain tissue section, and this is how it looks like. So we used here a particular method called 4I, and this is a chronic active lesion. You see here the lesion center. Here you see the lesion rim, and here you see the normal appearing white matter. And for aficionados, this is really a post-demyelinating lesion because you see a lot of macrophages still in the lesion center. So we stained for these different markers in this example. So these are cell type specific markers, but also activation markers for astrocytes and for microglia cells. And this here is a magnification. You can zoom into this, and these are of course processed images. So the cells, they were outlined, and you can see that you can see the individual cells, and you can see that they express different amounts of the tested proteins, and you can also see that some cell types interact and others don't. So these images, they require a lot of computational analyses, and here are some of the tools of the computational pipeline that we applied. When you do this computational analysis, you can cluster individual cells, just like with RNA-seq, and we did that here. And you can see that we clustered 6 different astrocyte subpopulations. And I want to direct your attention to the two different astrocyte populations that express A2A receptor. One of them is also characterized by high expression of GFAP and complement component 3. The other A2A high population expresses many markers, so we refer to that as pan-activated astrocytes. So we can now project these phenotypes, these clusters, back onto our lesions, and here's an example. So you see here a chronic active lesion. This is the center, this here is the rim, and this is the normal appearing white matter. And I want you to focus on these GFAP high, A2A receptor high astrocytes in cyan, and you can see that they're really present at the lesion perimeter in the normal appearing white matter. The pan-activated astrocytes are present in the lesion rim and also in the normal appearing white matter. And we quantified that here. So gray indicates normal appearing white matter, and red indicates the lesion rim. And I want to just show you that the other phenotypes that we identified, for example, a quasi homeostatic phenotype is present predominantly in the lesion rim, in the lesion center here in blue. And another phenotype characterized by high YKL40 expression is present in the so-called juxtavascular space. So this is the space around blood vessels. And you can do then a phenotype transition analysis called fate mapping. And you can see that the initial cells are really these YKL40 astrocytes in the juxtavascular area, and they transition into an intermediate phenotype, these A2A receptor high astrocytes, and those then transition into these homeostatic astrocytes in the lesion center. And you can show that in the schematic here. So the lymphocytes extravasate. The astrocytes that encounter, the first astrocytes that encounter these lymphocytes are these YKL40 lymphocytes here, and they develop into A2A receptor positive astrocytes, and those develop into homeostatic astrocytes in the lesion center. So this adds some context to where the A2A receptor expression plays a role. We then wanted to know, well, what does A2A receptor signaling do to astrocytes? And first we found that the ADORA2A expression, so the gene for this receptor is highly upregulated with stimulation with interleukin-1 beta and TNF alpha, and that stimulation of this receptor leads to upregulation of protein kinase A and into phosphorylation of CRAB, which is the transcription factor. So we verified that the CMP signaling pathways is activated through this receptor. We then looked at differential gene expression in stimulated astrocytes that were treated with an A2A receptor agonist and were not treated with this agonist. And we did then a differential pathway analysis and a gene set enrichment analysis. And the theme for all these analyses is the same, and that is that tight junction molecules and cell adhesion molecules are downregulated. You can see that here in the pathway analysis. So cell extracellular matrix interactions are downregulated, cell junction interactions are downregulated, cell-cell communication is downregulated. You see that also here. All these pathways here associated with cell adhesion are downregulated. Inflammatory responses were generally downregulated with the exception of IL-23A, which was upregulated. And here, as in sanity check, we see that CMP-mediated signaling is upregulated, which is what you expect. And we showed then also in these cell cultures that, in fact, Claudin-4, for example, which is a tight junction protein, is downregulated. And we showed that EphA2 and beta-catenin were all downregulated. So what does this now do to astrocytes? We found that treating with an A2A receptor agonist reduced the connectivity and the adhesion of astrocytes. So they balled up, and this is something that you can see here. So the extracellular space between the astrocytes was increased, and we quantified that, and you can see that the A2A receptor treatment makes a big difference. It greatly increases the extracellular space. When we did a trans-astrocyte migration assay with lymphocytes, we found that the lymphocytes that were put on top of a layer of astrocytes treated with an A2A receptor agonist, that this resulted in increased migration of lymphocytes through this layer. We also found, because it is known that A2A receptor mobilizes intracellular calcium stores, we looked at calcium signaling, and we found that calcium signaling was quite disturbed. So we found there was a great increase in spontaneous calcium spikes, and we found that there were these spontaneous calcium waves affecting other astrocytes. So there's some hyperactivity with regard to calcium signaling, both on a cellular level and on a network level. We also looked at glutamate transporters, and we found that the glutamate transporter GLAST was downregulated. And both this aberrant calcium signaling and the downregulation of GLAST suggest that there would be increased extracellular glutamate. And we see that this is in fact the case, that A2A receptor signaling leads to upregulation of extracellular glutamate, which again might cause glutamate excitotoxicity. And finally, we found that IL-23 secretion is also upregulated. We then went back to our brain tissue and we correlated A2A receptor expression with expression of different proteins. So for example, we correlated pCREP and A2A receptor expression, and there was a strong positive correlation indicating that if there is A2A receptor expression, there's also A2A receptor signaling. Then we found that adhesion molecules were downregulated, so they were negatively correlated with A2A receptor expression. This was true for Claudin-4 and for EphA2. We also found, just like in cell culture, that the astrocyte footprint was reduced. And you can see that here in an extreme example here and here. And we found that the immune cell infiltration was positively correlated with A2A receptor expression. We found that IL-23 expression was upregulated, but for GLAST, we found that there was no correlation between GLAST expression and A2A receptor expression. So to summarize this, with PET imaging, we showed that a secondary progressive MS is associated with high perilesional A2A receptor expression. In histology, we showed that there is one particular phenotype characterized by high expression of GFAP and A2A receptor that is present in the normal appearing white matter. This is an intermediate phenotype. In primary human astrocytes, we showed that A2A receptor expression is increased with stimulation, and that A2A receptor signaling reduces adhesion molecules, astrocyte connectivity, astrocyte footprint, increases lymphocyte migration, increases IL-23, and increases aberrant calcium signaling, reduces GLAST, the glutamate transporter, and increases extracellular glutamate. And we then went back to our MS tissue and we found that there are some correlations. That is, A2A expression correlates with phosphorylated CREP, with the transcription factor, that it downregulates adhesion molecules, and that the astrocyte footprint is reduced, and that there's increased immune cell infiltration and increased IL-23 associated with A2A expression. So to and these are the last slides. Just
let's finish up.
Okay, so just to summarize, we think that A2A receptor signaling disrupts the astrocyte network, which leads to increased infiltration with immune cells and perhaps to neurotoxicity. If we would block A2A receptor signaling, then we would restore astrocyte connectivity, and that might restrict the inflammation. I wanted to say that we have now generated inducible astrocyte-specific ADORA2 knockout mice, and our first experiments show that the severity of chronic EAE is in fact reduced. These are very preliminary results, and we certainly haven't done any histology. I want to mention again that this drug that is FDA approved for Parkinson's disease, istradefylin, we're giving this now to these mice and see whether that makes a difference. And if these mouse experiments pan out, then there might be an argument that we can use, in fact, this drug for MS patients. And I'm closing with acknowledging.
Okay, David. Thank you. We're going to stop here. Okay, David, thank you very much. I'll introduce Tomo now. Okay, next is Professor Tomokazu Sumida, who's going to talk about some very new unpublished data related to regulatory T cells.
All right, can you hear me?
Yes.
Okay, yeah. Thank you so much for the introduction, David. I'm also really thanks for the organizer to give me this opportunity to present my work today. Today, I would like to talk about the potential molecular mechanisms that can induce the loss of peripheral T cell tolerance in MS. So here are the contents that I would like to go through with my today's talk. But most of the background has been already described by David Hafler and then David Pitt. So I'm gonna go quickly for that. But then I'm gonna show some transcriptional profiling of human T cell, especially for the Treg, and also share the novel identification of epigenetic profiling of those cells. Then finally, I will show some potential link between genetic susceptibility of MS and gene regulatory circuit that I found through this transcriptional and epigenetic exploration. So as we all know, human genome encodes the blueprint of the phenotype. How this genomic code translated into the phenotype is represented by multiple steps, epigenetic and transcriptional and proteomics. Therefore, the understanding of this genetic code is very important, very fundamental. How this MS phenotype as a MS could be driven by genetic code. So what we know so far about the MS genetic information. As you have already seen this figure, the decade of effort centered on IMSCC identify these 233 MS-associated risk variants. But through this large cohort of MS-GWAS study, only 22% of MS risk can be explained by just genetic variants. That being said, the other 80% of risk could be explained by as a result of combination of environmental factors and genetic factors. This fact highlights the importance of understanding how epigenetic and transcriptional changes are modulated in MS, because those are the factors affected by environmental stimuli. So Roadmap and ENCODE project have advanced our knowledge of the functional elements in the genome and epigenome. Around 90% of autoimmune susceptible loci has been mapped in non-coding region, suggesting they are affecting the regulation of gene expression rather than the function of the gene itself or a gene product. This heat map has already shown by David, but again, this MS-associated genetic susceptibility loci have highly enriched in the genetic regulatory reason from autoimmune, from T cell and B cells, especially for including Treg. Moreover, current advancement of single cell or single nuclear RNA-seq provides the further evidence that a lot of EQTL effect of genetic variants is cell type specific. So the question is, what is the cell types relevant to MS pathophysiology? Accumulated evidence suggests that MS is an autoimmune disease driven by autoreactive CD4 T cells, and loss of peripheral tolerance, such as Treg dysfunction, is one of the features of MS. So based on this theory, hundreds and thousands of studies has been conducted focusing on the CD4 T cell function in the context of MS. So what we know about CD4 T cell in MS, people start to investigate the transcriptional profile of total CD4 T cells isolated from MS patients. So here are the two study attempt to identify the transcriptional difference between healthy and MS patient in total CD4 T cell. However, their attempts have ended up with failure to detect significant difference in gene expression. As CD4 T cell display a large degree of functional diversity, the interrogation of total CD4 T cell as a bulk population did not allow identification of causal transcriptional changes that you can predict to see in MS T cells. Single-cell RNA-seq data that you can see here can identify two major axes within CD4 T cells. One is conventional T cell, and another is conventional T cell versus regulatory T cells, and another one, is the naive and memory axis. So we need to examine those at least 4 subsets of CD4 T cell independently. However, the epigenetic and transcriptomic changes of these CD4 T cell subpopulation has not been fully explored. So today, the question that we are trying to address in this project is, what is the gene regulatory program that is altered in MS and that can drive dysfunctional properties of MS T cells? The method that we are using for this project is bulk RNA-seq and single-cell RNA-seq based transcriptional profile of CD4 T cells, and epigenetic profiles of CD4 T cell using the bulk RNA-seq, bulk ATAC-seq, and finally functional assay for validating those factors that is identified by transcriptional and epigenetic profiling by using the human primary Tregs. Here is the overview of the experimental workflow. In the sake of time today, I'm mostly focusing on the analysis for memory cell, especially for memory Treg subset, because the interesting difference was observed in this memory Treg population. Here is the volcano plot showing the differentially expressed gene in memory T conventional and memory Tregs between MS versus healthy. When we look into the genes that are upregulated in both Treg and T conventional cells in MS, the PRDM1 came out as top transcription factor upregulated in MS. This result was confirmed in validation cohort, which is completely separate from the discovery cohort using the qPCR. We also confirmed this upregulation of PRDM1 by using the flow cytometry at protein level. We tried to validate this result by using the single-cell RNA-seq. So, but not only just messenger RNA, but also the site-seq that can identify the cell surface immune molecules on top of RNA expression. We were able to determine not only memory Treg, but also T helper subset within the memory Treg, such as Th1-like, Th17-like, Th2-like Tregs. So as you can see in this plot, which is the data not from the flow, but also the site-seq, showing the CXCL3, CCR4, CCR6 expression within the memory Treg subset. And we were able to distinguish Th1, Th17, Th2-like subset within this Treg. Then the PRDM1 upregulation MS by identifying this by the single-cell RNA-seq was identified as a top upregulated gene, which is consistent with bulk RNA-seq. But more interestingly, this PRDM1 upregulation was highly enhanced in this Th17-like Treg subpopulation, which cannot be identified by just bulk RNA-seq. In other words, we got this information because of the advantage of single-cell RNA-seq using the site-seq. We also checked the transcriptional profile of memory Treg from the ImmuneNext UT database, where 28 immune cells, including memory Tregs from 10 immune-mediated disease are analyzed. Of note, PRDM1 upregulation and some of the genes expression signature that we observed in MS Treg seems to be a common feature among Treg in multiple autoimmune disease, as you can see here, especially for the SLE or type 1 diabetes. So this indicates our observation looking at the PRD1 upregulation in MS Treg is not unique to MS, but potentially common feature across multiple immune-related autoimmune disease, where you can see the dysfunction of Treg. Okay, so the question is, what is the function of PRD1 in Treg? When we look into the literature, we were puzzled because previous mouse study demonstrated that PRD1 plays an anti-inflammatory role in Treg through maintaining the FOXP3 expression. And then more specifically, the Treg-specific PRD1 knockout mice displayed exaggerated EAE phenotype, indicating the PRD1 is necessary to prevent EAE development. This is clearly contradictory to what we observed here. I mean, increased PRD1 expression in dysfunctional MS Treg. So this is the kind of the issue that we have been struggle for like a few months. But then one day, my collaborator at Manoj Kelly's lab at Broad Institute noticed our sharp peak located at PRD1 locus with his Hydra analysis. So let me explain very quickly about this Hydra analysis. Hydra is the novel method developed by their lab, Kelly's lab, to identify the highly active promoter or enhancer elements. Therefore, this Hydra peak, you can see here, falls into the promoter or active enhancer. And then this made me realize that this PRD1 has short isoform generated by alternative promoter usage, as you can see here. So what is this short PRD1 isoform? Short PRD1 lacks the N-terminal region, and which results in missing a part of PR domain that is important to mediate interaction with cofactors of PRD1. Therefore, the short PRD1 is important to show their unique function compared to the full-length PRD1. We checked expression of short and long PRD1 isoform in multiple human immune cells and found that this short isoform is dominantly expressed in memory T cells and NK cells. In contrast, the monocyte and the B cell subset preferably express the long PRD1 isoform than short isoform. Then we check the short and long PRD1 expression in our bulk RNA-seq data and identify the our original finding showing the upregulation of PRD1 is mainly driven by this elevation of short PRD1 isoform you can see here. Another note is that this short PRD1 isoform is created during the dry nose primate evolution. So mouse genome doesn't code this short isoform. So this is unique in humans, in primates. Okay, so next we try to address these questions. What is the downstream factor induced by PRD1 short isoform? And also, does increased PRD1 short isoform drive Treg dysfunction? To assess the direct impact of short PRD1 on the human Treg, we transduced short PRD1 into human primate Treg by using lentiviral transduction. We found SGK1 as one of the top upregulated gene by short PRD1 overexpression. And then we confirmed this SGK1 was uniquely induced by only short overexpression, but not long PRD1 overexpression. The role of SGK1 in Treg has been already shown by us and the other groups. Treg-specific SGK1 knockout mice develop less severe EAE, and high salt-induced Treg dysfunction is reversed by SGK1 inhibition, indicating that this SGK1 limits Treg suppressive function in the context of MS and autoimmune disease. Next, we confirm if the short period overexpression impairs Treg function or not. And we performed in vitro Treg suppression assay, and as you can see here, short period overexpression suppress Treg suppress function, inhibit the Treg suppression function. So clearly, this short period induced Treg dysfunction. And this short period overexpression also decreased the FOXP3 protein stability. That can lead to this Treg dysfunction. Okay, so let's shift gears to epigenetic profiling. To further understand the upstream mechanisms that control gene expression, in our case, what is the upstream regulator of PRDM1? We focused on epigenetic alteration. We adopted ATAC-seq, a technique that allows us to identify the open chromatin region at a genome-wide scale. So ATAC-seq has advantages over conventional method because ATAC-seq needs lower inputs, and then whole procedure is simple and then quicker. So then ATAC-seq can give us this open chromatin region where the transcription factor can bind to the DNA and they induce the downstream gene expression. By using this ATAC-seq data, we can further analyze this footprint analysis or motif analysis to identify exact transcription factor that can bind to the DNA on open chromatin region. And then by using the ATAC-seq, we asked if there is a difference in chromatin accessibility between MS and healthy Treg first. But unexpectedly, we could not detect significant difference between MS versus healthy Treg in terms of chromatin openness. So this indicates that the chromatin openness or chromatin structures of MS Treg are kind of similar to that of healthy control. How can this be explained? Here is our hypothesis. Even the regulatory elements, open chromatin regions, are similarly open. Depends on the transcription factors that bind to the regulatory region, the impact on PRD1 induction could be different. As like this. So to determine transcription transcription factor binding on accessible or chromatin region, we perform footprint analysis. Footprint analysis can tell us the transcription factor that actually binds to the DNA. So then we can get more precise information compared to the motif analysis. But to perform footprint analysis, we have to have deeper sequencing of ATAC-seq data. Then we sequence deeper and perform footprint analysis. Here's the result. And we found that AP1 transcription factor highlighted in red box are highly enriched in MS Treg. This indicates AP1 transcription factor is prime in MS Treg DNA, open chromatin region. So what is the fundamental pathway that can activate AP1, which is combination of T cell receptor and CD28 co-stimulatory signaling? Recent finding also highlighted that this AP1 binding are highly enriched in MS MS susceptible loci. Importantly, the CD28 locus harbors multiple EQTLs. CD28 locus harbors multiple EQTL loci and associated with multiple autoimmune disease, including MS. Given the tightly linked CD28 signaling in AP1 activation, MS GWAS risk variance near CD28, this RS12614091, could be a potential explanation why this prime AP1 binding that we observed in MS Treg is associated with MS. Of note, when we look at the gene expression pattern of AP1 family and AP1 family transcription factor and CD28 in our Treg transcriptional data, we found very strong positive correlation between those CD28 and AP1 transcription factor genes and short PRD1 expression, specifically in MS. This suggests that AP1 enrichment is linked with short PRD1 induction in MS Treg. But there is no direct evidence how this AP1 can induce a short PRD1. So we sought to address this question, what is the gene regulatory mechanisms that directly induce short PRD1 upregulation? To address this question, I first determined the candidate regulatory elements, open chromatin regions, nearby PRD1 locus, and then adapted the CRISPR-A system to identify the regulatory elements that can induce short PRD1 induction. We established CRISPR-A system in Jurkat T cells so that we can examine which regulatory elements have a potential to induce short PRD1. And we assessed total 20 top candidate chromatin accessibility loci by using this system. Within this 20 loci, we found one peak that most potent regulatory element that induce short PRD1 expression. When we zoom into this region, actually the AP1 DNA binding motif and AP1 ChIP-seq peaks are highly uniquely enriched in this peak region, suggesting that AP1 binding could confer the short PRD1 upregulation in MS Treg. All right, to summarize this, we all know that the TCR, we found this AP1 priming and short PRD1 induction was observed in MS Treg. And this short PRD1 further upregulates the downstream SGK1, which is known to and also induce FOXP3 destabilization. Both SGK1 and FOXP3 destabilization is known to link to Treg dysfunction. And from conventional immunological aspect, CD28 signaling and TCR signaling is known to induce this AP1 binding activation. And this could be explained by known well-established eQTL effect of CD28 locus in MS, which can enhancing the CD28 signaling, resulted in AP1 priming on Treg DNA, Treg chromatin open DNA region that could induce short PRD1. Finally, this SGK1 induction and then Treg dysfunction has been known to induced by high salt environment that we have been studying so far. So we are trying to link this genetic susceptibility to epigenetic and transcriptional modification of MS, whether it's affected by this high salt environment or not, as a future direction. With that, I would like to thank David Hafler for his great supervision and mentorship, and also all the lab members in Hafler lab, especially the Matt, for this project. Also, this work has been done through tight collaboration with Manolis Kelly's lab, especially Lian and Yonjin, and Brock Bernstein lab and Czech. And the eQTL data and transcriptome data for the other autoimmune disease are achieved through the collaboration with Dr. Ota and Fujiu at the University of Tokyo. With this, I would like to finish my talk and happy to take any question in the discussion section. All right. Thank you so much.
Thank you, Tomo. Back on time. Okay. Next is Professor Erin Longbrake, who is in the also Department of Neurology at Yale and is leading our head of clinical trials in the Neuroimmunology division. Erin, you're on.
Great, thank you so much for the invitation to talk today. And I'm going to take things back up to the macro level, away from the micro level. We're going to talk about clinical trials and multiple sclerosis. And in the spirit of the session of protecting the brain, we're going to focus on very early multiple sclerosis. So I'm going to start this off by telling you stories about two individuals. One was a 35-year-old professional who first had symptoms of multiple sclerosis when he was 24 years old. And he was at the time taking a major test and had complete vision loss, which then resolved over 1 to 2 days. And he was busy, did not have time to deal with this. And so he did not. Felt back to normal and went on with his life. 5 years later, when he was 29, he experienced some mild to moderate dysarthria for 2 weeks and some numbness on his left side, which again, he chalked up to stress and did not seek medical care. And the symptoms once again resolved. When he was 31, so again, another 3 years, he felt, had recurrence of the left-sided numbness, and now with some clumsiness and inability to walk straight, which again was short-lived and he didn't seek care. And then I finally met him when he was 35. At that time, he had had the emergence of left-sided ataxia. He was really unable to walk for any duration of time and had extreme fatigue that was limiting his ability to get through a day. MRI of the brain revealed many lesions, many of which were contrast enhancing. And of course, we diagnosed him with multiple sclerosis. At that point in time, he had had symptoms for well over 10 years. And finally was getting this diagnosis. The other story that I'll tell you is about the same age, a young man who was a first responder. He came to the clinic complaining of a pressure-like sensation behind his left eye, which had been worse with eye movement for about 2 weeks. There was really no loss of vision, and the symptoms were minor, but was enough that he felt like, oh, you know, I'll get it checked out. We did an MRI of this gentleman as well, and he also had lesions that were characteristic of multiple sclerosis, and we were able to make a diagnosis of MS in this gentleman as well. Both of these patients initiated treatment with the same medication, ocrelizumab, promptly at the time that they were diagnosed. And ocrelizumab is one of the most effective medications available for relapsing multiple sclerosis. So this was a very high efficacy agent. It was promptly started at the time of diagnosis, but the outcomes were not the same. The first patient did not have resolution of his fatigue. His fatigue became disabling and he had to ultimately reduce the hours that he was able to work. His left-sided ataxia and leg weakness continued to be present, emerging with any physical exertion, and he was not able to really exercise strenuously or do anything that required a lot of physical exertion. And he ended up needing a cane for ambulation, mainly towards the end of the day or in situations where he was going to be on his feet for a long time. The other patient's visual disturbance resolved completely over a few days. Indeed, it was resolved by the time I saw him initially in clinic. And since then, he's had no additional manifestations of multiple sclerosis, either clinical or on MRI. So why were these outcomes different? What was the difference between these two cases? Here you see an illustration of multiple sclerosis presented as a continuum. With the first event suggestive of multiple sclerosis, we call that a clinically isolated syndrome. So it's the first clinical presentation. The disease tends to be quite inflammatory early in the disease course. So you can see illustrated by the purple line that early in the disease, there's a lot of inflammation, breakdown of the blood-brain barrier, inflammatory infiltrates into the brain. And that tends to be concentrated in the early days of the disease. The brain volume tends to gradually get lower over time. And so early in the course of multiple sclerosis, the brain volume is on par with others of the same age, but the rate of brain shrinkage is more rapid in multiple sclerosis. And so as the disease progresses, these brains start to have neuron loss and shrinkage. And so the nerve cell loss increases with increasing time. Our first patient was diagnosed relatively late in the disease. So he was 10 years at least into the pathophysiologic course of multiple sclerosis by the time he was diagnosed. And so he was later in his disease course. Patient number 2, in contrast, came to medical attention with the first clinical attack. So it would not have been possible to diagnose him any earlier than he was diagnosed. And that, at the heart of it, is the fundamental difference between these cases. We know that treating multiple sclerosis early gives better results. There's been quite a few trials now where the clinically isolated syndromes were compared to patients who started treatment later in the course of the disease. And as in a somewhat unusual situation for medicine, it was a very clear answer. Every one of these trials showed the same information. The patients always did better. They had fewer relapses, ultimately had less disability when the treatment was initiated early. And that was true no matter what the treatment was. So here I've listed 5 different types of disease-modifying therapies for multiple sclerosis. Every single one of these trials had the same result, and that was that patients who initiated treatment at the stage of a clinically isolated syndrome ultimately did better over the long haul in their disease. So that I'll illustrate here. When one starts treatment late in the course of disease, I'll liken that to standing halfway up a mountain while an avalanche is coming down at you. By the time you bring the immunomodulatory therapy on board, there's been years of a dysregulated immune system and the resultant underlying neurologic injury. And the hope of reversing that is very similar to your likelihood of stopping the avalanche by standing halfway down the mountain. It's not going to happen. And so in a sense, treating late in the MS disease course is a missed opportunity that we don't expect at that point in time that we'll be able to completely reverse the dysregulated immune system and the resultant damage to the neurologic system. So this all comes down to our ability to diagnose multiple sclerosis. And we've heard a lot about the biology of multiple sclerosis and how the immune changes emerge. But clinically, when we tell someone you have multiple sclerosis, we are relying on 2 things. One is called dissemination in space, one is dissemination in time. And essentially what that means is they have to have multiple lesions within their central nervous system that are in distinct locations. And likewise, those lesions have to have developed over more than one period in time. The dissemination in time and space is the hallmark of a clinical diagnosis of multiple sclerosis. There's 4 pathognomonic locations within the central nervous system. One is juxtacortical, or touching the cortex. One is periventricular, or touching the ventricles. One is infratentorial, or within the brainstem or cerebellum, and one is in the spinal cord. And to prove dissemination in space, the individual has to have at least 1 lesion in at least 2 of these 4 locations. At the end of the day, making a clinical diagnosis of multiple sclerosis requires symptoms. So even if one has lesions, even if one has abnormalities, it's by convention, we have decided that multiple sclerosis requires symptoms. And so that can be optic neuritis or loss of vision, could be transverse myelitis caused by inflammation in the spinal cord, or more generally weakness, some sort of sensory disturbance or ataxia, brainstem syndrome, or any number of others. But at the end of the day, there has to be a distinct clinical episode that brings someone to medical attention. And we can't really make a diagnosis of MS in the absence of any corresponding symptoms. So I'm gonna just illustrate this in a different way. So multiple sclerosis biologically and multiple sclerosis clinically are not quite talking about the same thing. So David and Tomo all talked to us a lot about the biology of multiple sclerosis. And that, I think, is going to be extremely important. But clinically, the phenotypic spectrum, what does someone with MS look like? It exists on a spectrum, and I'm illustrating this as a bell curve. On one side of the spectrum, we have individuals who have very severe disease, who have a relentlessly progressive course, or perhaps they're using wheelchairs within a few years of the time of their diagnosis, patients who are tremendously disabled. On the other side of the spectrum, you have people who live their day-to-day life with really no symptoms of multiple sclerosis, and this may be even people who are untreated. In fact, the most striking illustration of this other end of the spectrum is the well-established phenomenon where people will go to autopsy, and at autopsy, they are discovered to have demyelinating changes in their brains, which looks exactly like the multiple sclerosis lesions would look. But the disease in those patients never caused any symptoms that brought them to medical attention. And so there's a full spectrum of disease. And so we say that the clinical symptoms are going up over the course of this phenotypic spectrum. The further on this side you are, the more symptoms you're going to have. The further on this side, the fewer symptoms. And there's a certain symptomatic threshold that you have to hit before the disease would come to medical attention. You wouldn't be given a formal diagnosis of multiple sclerosis unless or until the symptoms that one lived with were enough to pass that threshold. And so everything on the left side of this curve is therefore would never necessarily come to attention, would not be described as multiple sclerosis in the clinical practice, even though the biology of MS may be working underneath the surface. And so here, again, on the right side, we have people who meet MS diagnostic criteria. They have MS. I'm really interested in this left side, though, because this may be a preclinical form of the disease for many patients. And so there's two illustrations we're going to use for this asymptomatic side of things. One is the phenomenon of radiologically isolated syndrome. So what is radiologically isolated syndrome? So I'm going to show you a few cases. This picture, you will note, looks strikingly similar to the MS pictures I showed you earlier. This very characteristic ovoid lesion by the ventricles is highly suggestive of demyelination. And there's also some juxtacortical lesions here. This is a woman whose MRI was done, however, for just non-specific mood problems, nothing that would have bought her a diagnosis of MS. And in the course of her workup, her spinal fluid was noted to be positive for oligoclonal banding, suggestive that the immune system was more active around the brain than elsewhere in the body, which is, again, highly suspicious for an underlying inflammatory immune disease like multiple sclerosis. Here's another case. You can again see these paraventricular lesions, juxtacortical lesions, and this individual was a 60-year-old man whose MRI was done for headaches. And once again, it was highly suggestive of multiple sclerosis. If he had had the right clinical situation, he would have met characteristics, but he did not. The headaches are not considered, generally speaking, to be a syndrome that's suggestive of MS. So we call both of these individuals as having a radiologically isolated syndrome. And RIS is diagnosed when there is neurologic damage apparent on the MRI. And indeed, these MRIs can meet diagnostic criteria for multiple sclerosis in terms of dissemination in space. Some RIS patients may have evidence of inflammation within their CNS in that they may have oligoclonal banding, and they may also have evidence of axon damage evidenced by the presence of elevated levels of neurofilament light chain in their blood. But yet they've not had any episodes definitively suggestive of multiple sclerosis and hence do not meet clinical diagnostic criteria for MS, even though some of these individuals probably have biologic MS and are just on the left-hand side of that bell curve. So our hypothesis then is that pathologic immune shifts coincide with radiologically isolated syndrome and probably also precede it. And so we would add an immunologically isolated syndrome as probably the first step in the pathologic cascade that ultimately leads to multiple sclerosis, followed by a radiologically isolated syndrome, and then ultimately the passing of the clinical symptomatic threshold and a diagnosis of formal multiple sclerosis. For many, RIS precedes multiple sclerosis. The data here are 10-year observational data of individuals diagnosed with a radiologically isolated syndrome. And you can see that at 5 years after the RIS was identified, about 1/3 had clinically converted to definite multiple sclerosis. And about 10 years after the diagnosis of RIS, half of the individuals had definitively converted to multiple sclerosis. Now, the flip side of that is that at 10 years after the identification of the lesions, half did not have MS. And so that we have to keep in mind as well. Other features allowed us to stratify. Patients were more likely to go on to have MS when they had either spinal cord or infratentorial lesions present, when they were younger at the time of the diagnosis of RIS, and when they had oligoclonal bands present in their spinal fluid. Again, that was a risk factor for future multiple sclerosis. So in summary, multiple sclerosis, clinical multiple sclerosis has a preclinical prodromic phase, which is associated with potential radiologic changes as well as immunologic changes. Many of these patients in this prodromal phase will go on to have multiple sclerosis in the future, but there are not any high quality data to tell us how we should treat these individuals while they're in this preclinical phase. And in fact, most providers would not institute immunotherapy until the onset of clinical symptoms, and certainly no insurance company would pay for treatment until the onset of clinical symptoms. Is that the right answer though? Well, we don't know yet, but there are some clinical trials designed to help us address this. So the idea with treating multiple sclerosis at the preclinical or prodromic phase, going back to my avalanche illustration, in this sense, perhaps we're standing closer to the top of the mountain or at the top of the mountain. The immune system dysregulation is just getting underway, and the underlying neurologic injury is still at a minimum. In this situation, could immunomodulation prevent the disease so that the clinical signs of disease never become evident? That is the million-dollar question. So there's two ways where we can kind of, okay, what's the best we can do currently with detecting multiple sclerosis in people? The first is the first demyelinating event. And I already shared with you the data that says that treating at the first demyelinating event is better. But what if we move earlier? What if we move into the preclinical prodromic period and treat at the radiologically isolated syndrome stage? Well, there are a few clinical trials designed to do this. First, I'm going to talk about two trials that are initiating currently approved MS treatments that are designed to be given as a lifelong immunotherapy. The first is the TERRACE trial, which is using teriflunomide versus placebo in individuals with radiologically isolated syndrome. This is a randomized, double-blind, placebo-controlled clinical trial enrolling 125 patients and following them for 2 years, the primary outcome being a first demyelinating event, or that first clinical syndrome suggestive of multiple sclerosis. The TERRACE trial is fully enrolled, and they anticipate completing the study late this year. However, the results are not yet presented or published. Similarly, the ARISE clinical trial is using dimethyl fumarate. This is a twice daily medication for works for multiple sclerosis. They're comparing it against placebo for radiologically isolated syndrome. Once again, this is a randomized double-blind placebo-controlled trial, enrolling just short of 100 patients, following them for 2 years, with the outcome being a first demyelinating event. This is fully enrolled. Results are not published, but some preliminary results were posted on clinicaltrials.gov, and it looks like it's probably going to show benefit in preventing demyelinating events in this group, but the full data have not yet been presented. Both of those medications are ones that you have to continue taking the medications in order for them to work. So once you stop them, they'll stop working. And so an alternative approach to treatment would be to use a short induction therapy, where you give an immunomodulator for just a short period of time and then stop it, hopefully in so doing, resetting the immune system back to a better, less pathologic state. And one potential medication that we are interested in using as an induction approach is using ocrelizumab. Again, ocrelizumab is a B-cell depleting medication that induces global B-cell depletion of mature B cells, but not plasma cells. And we are hoping that by deleting that from the lineage, we may induce long-lasting effects. B cells did seem to be more and more likely to have an early impact in the pathophysiology of multiple sclerosis. And and ocrelizumab usually it is approved for multiple sclerosis. It is given continuously in multiple sclerosis, but it's been demonstrated there are long-lasting immune effects. Just how long acting is not yet known, and whether those would be sufficient to prevent future disease if it was given early enough in the disease pathophysiology is also not yet known. So at Yale, we are partnering with Genentech to do an investigator-initiated clinical trial we call CELLO. And in this study, we are taking people with radiologically isolated syndromes. They are randomized either to placebo or to ocrelizumab for just 3 full courses or 18 months worth of B cell depletion. And the individuals in the study will be followed for a total of 4 years. We are immunology heavy, as may not surprise you from coming from our group. In addition to the traditional clinical and MRI assessments, we'll be doing flow cytometry of blood and CSF, as well as single cell RNA sequencing of blood and CSF immune cells, with our goal being to really get at the very earliest immune changes that are associated with disease. Some unique features of the CELLO trial is this short-term use of immunotherapy and induction approach, which may be much more desirable to individuals with RIS who, recall, have no symptoms of MS. And so asking them to take medicines that may have side effects and will suppress the immune system for the rest of their lives is a pretty big ask. Whereas a short-term immunotherapy approach might be more acceptable. The CELLO trial is also recruiting people with a lower disease burden. And so hopefully these are people who are earlier in their disease pathophysiology. And this is different from the other 2 RIS trials. There are some slight differences in the inclusion criteria, but at the bottom line is that the people who are enrolled in CELLO may have less radiologic disease than those who are in the other 2 trials. CELLO is also following patients for longer than the other studies and has much more of an emphasis on biomarkers and the immunobiology of very early MS. To help bring people into the trial, we are also instituting a screening program for close family members of MS patients. This is a multi-centered study. It is US-based, and we have about 16 centers across the US. We are hoping to learn from this study whether ocrelizumab can prevent clinical symptoms of MS and or new radiologic damage associated with MS in an at-risk population who do not yet have clinical disease. And we're also hoping to understand whether transient or short-term B cell depletion is an effective immunomodulatory strategy in this group. On the immunologic side, we are hoping to be able to describe gene expression changes and immunophenotype shifts that characterize the emergence of clinical multiple sclerosis as it evolves, and also to characterize the reconstituting immune system after B cells have been temporarily depleted. We have a website for the study at cellostudy.org, so many more details are available there if anyone has interest. And the study protocol has also just recently been published in Multiple Sclerosis and Related Disorders. So in conclusion, the immunotherapies tend to have much more clinical effect when they are instituted early in the disease pathobiology, which begins well before the first clinical symptoms of disease. We are occasionally fortunate enough to discover multiple sclerosis at a preclinical stage, and clinical trials are ongoing for this radiologically isolated syndrome to determine if early enough intervention could prevent clinical signs of multiple sclerosis. And ongoing research is desperately needed in order to be able to systematically identify these at-risk people rather than relying on an MRI that was serendipitously discovered to have lesions. So better ways to find these patients are needed. And with that, I will conclude, acknowledging the MS group at Yale, as well as my clinical trial group and my collaborators at Genentech who are so instrumental in working with us on the CELLO study. And that concludes my talk. Thank you so much for your attention.
Thank you. Yeah, I would just ask for the individuals at Prada. David just wrote that people tried to log in from Yale and the login didn't work. I wasn't aware of that. we don't have any other participants. So that's beyond our control. So I don't know if we have any questions for each other, but we could also just end now. So I'll ask the people from preserving the brain to step in and let us know what we should do. Hello? Okay. Well, if no other questions, I think everyone kept on time. Thank you very much. And I think we have questions to each other. So let me ask if any questions to each of the panelists. We can do that. This is going to be streamed by YouTube. So Dave, all your wonderful students can watch us then. So questions. Hello? So I'll make a comment to David. So David, we had these discussions. So we showed our data from the FAR paper showing that the hits with the genetic variants were predominantly, were hitting immune cells and not affecting the brain. But as David pointed out, the flaw in what we did in that paper was really a first generation, was there's part of the ENCODE, the NIH-funded ENCODE project. And we took brain, or they took brain, put in the blender. And that's how the DNA was identified. So David's study really, and it shows how as we gain more knowledge, one can better fine tune the different experiments. And I think by using the astrocytes that David did was a brilliant way of going deeper, because astrocytes are kind of like an immune cell at one level, and we have many hits to microglia. So it points out to where we are with all these data. I also would make a comment, with Erin. I think one of the whole concept that the group has come up with, we talk about RIS, radiologically isolated syndrome, but the concept of immunologically isolated syndrome, that is a disease that begins before the parenchymal involvement, is critical in approaching autoimmune disease and MS. Unlike other common autoimmune diseases, the brain's kind of a special organ, and unlike the eyelids, or skin, or liver, a few lesions can heal and you're fine. Whereas in the nervous system, a few lesions can be devastating. So we want to be able to do is prevent disease. We don't know how to do it yet, but I will throw the gauntlet down to all those listen to help us develop ways of identifying patients who are in the process of developing MS, because before their parenchymal lesions, almost certainly there'll be inflammation in the meninges, which one sees in the EA model, and whether we could potentially identify those individuals before they begin to have parenchymal disease would be critical. And we don't know how to do that.
We're working mainly with people who have a pre-test probability, so are just at slightly higher risk compared to the general population. And so for now, we are focusing on building a cohort of close family members of individuals who have multiple sclerosis. And close family members certainly aren't guaranteed to get MS. It's probably maybe 5% in a close family member compared to 0.1% in the general population. But that's still we'll have some people who do go on to have a parenchymal disease, and family members also sometimes have brain lesions that can be suggestive. So Yale is working to build a cohort of close family members and to learn from them immunologically and radiologically. There's a number of other cohort studies of close family members around the United States and probably Europe as well. And so any close family members who may end up listening to this broadcast, maybe seek out a place where you can help us come up with these ways to discover, ways to prevent, ways to cure. We hope.
A question for Tomo. I think an absolutely fascinating observation with short form or period one. How do we make that into a therapy?
Yeah, that's a great question, and then also the tough question. So, so the long period one isoform is known to be really important to maintain the Treg fitness or Treg maintenance. So the way how to specify, interruption of short period one is critical if you wanted to target the period one. So we are trying to understand what kind of promoter specific for short period one is there or not. So then we can avoid detrimental effect of blocking period one that can potentially, induce these disruption of Treg homeostasis. But at the same time, this could be also applied to the cancer therapy potentially. If you can disrupt the Treg function in cancer, you can use that as a treatment. So there could be a lot of potential to try to tweak the PRD1 short and long balance to regulate the T cells. But at the same time, the short and long PRD1 isoform ratio is different, depends on the cell types. So we have to also take that into account.
I think that'll be important in an area to investigate. Okay, good. So I think we've gone through, it's now just about noon. So I want to thank all of you who will hear us on YouTube. We had a lot of fun listening to each other. And for those of you who live in Italy, I strongly encourage you to visit the Prada Foundation, the Preserving the Brain exhibition in Milan. It's just terrific. We made a video for it, and a lot of interesting overviews of the disease. So we invite all you to join and to join the exhibition. So everyone, take care. Bye.